सो हेलो गाइस, कैसे हैं आप लोग। माई सेल्फ सिद्धिकांत सिकवाल। और आज हम स्टार्ट करने वाले हैं बॉडी फ्लूइड्स एंड सर्कुलेशन एनसीईआरटी लाइन बाय लाइन। तो चलिएगा शुरू करते हैं बिना वक्त गवाए। You have learned that all living cells have to be provided with nutrients, O2, and other essential substances. Also, the waste or harmful substances produced have to be removed continuously for healthy functioning of tissues. It is therefore essential to have an efficient mechanism for the movement of these substances to the cells and from the cells. Different groups of animals have evolved different methods for this transport. Simple organisms like sponges and celandlates circulate water from their surroundings through their body cavities to facilitate the cell to exchange these substances. More complex organisms use special fluids within their bodies to transport such materials. Blood is the most commonly used body fluid by most of the higher organisms, including humans, for this purpose. Another body fluid, that is lymph, also help in the transport of certain substances. In this chapter, you will learn about the composition and properties of blood and lymph, that is tissue fluid, and the mechanism of circulation of blood is also explained here. 15.1 Blood. Blood is special connective tissue consisting of a fluid matrix, plasma, and formed elements. 15.1.1 Plasma. Plasma is a straw-colored viscous fluid constituting nearly 55% of the blood. 90 to 92% of plasma is water, and proteins contribute 6 to 8% of it. Fibrinogen, globulin, and albumin are the major proteins. Fibrinogens are needed for clotting or coagulation of blood. Globulins primarily involved in the defense mechanism of a body, and albumins help in osmotic balance. Plasma also contains small amounts of minerals like Na plus, Ca2 plus, Mg2 plus, HCO3 minus, Cl minus, etc. Glucose, amino acid, lipids, etc. are also present in the plasma as they are always in transit state in the body. Factors for coagulation or clotting of a blood are also present in the plasma in an inactive form. Plasma without the clotting factor is called as serum. 15.1.2 Formed elements. Erythrocytes, leukocytes, and platelets are collectively called formed elements, and they constitute nearly 45% of blood. Erythrocyte or red blood cells, that is RBC, are most abundant of all the cells in blood. A healthy adult man, on an average, 5 million to 5.5 million of RBC millimeter per cube of blood. RBCs are formed in the red bone marrow in adults. RBC are devoid of a nucleus in most of the mammals and are biconcave in shape. They have lead-colored iron-containing complex protein called hemoglobin, hence the color and name of these cells. A healthy individual has 12 to 16 grams of hemoglobin in every 100 ml of blood. The molecule play a significant role in transport of respiratory gases. RBCs have an average lifespan of 120 days, after which they are destroyed in the spleen, that is a graveyard of RBC. Leukocytes. Leukocytes are also known as white blood cells or WBC, as they are colorless due to lack of hemoglobin. They are nucleated and are relatively lethal in number, which average 6,000 to 8,000 millimeter per cube of blood. Leukocytes are generally short-lived. We have two main categories of WBCs, that is granulocytes and agranulocytes. Neutrophil, eosinophil, and basophil are different types of granulocytes, while lymphocytes and monocytes are the agranulocytes. Neutrophils are the most abundant cells, that is 60 to 65% of cells of total WBC, and basophil are least, that is 0.5 to 1% among them. Neutrophil and monocyte, that is 6 to 8%, are phagocytic cells which destroy a foreign organisms entering the body. Basophils secrete histamine, serotonin, heparin, etc. and are involved in the inflammatory reactions. Eosinophil, that is 2 to 3%, resist the infection and also associated with the allergic reactions. Lymphocytes, that is 20 to 25%, are two major types, that is B and T forms. Both B and T lymphocytes are responsible for immune responses of body. Platelets are called thrombocytes, are cell fragments produced from megakaryocytes, special cell in the bone marrow. Blood normally contains 1,50,000 to 3,50,000 platelets millimeter per cube. Platelets can relatively Platelets can release a variety of substances, most of which are involved in the coagulation or clotting of blood. A reduction in the a reduction in their number can lead to clotting disorders, which will lead to excessive loss of blood from the body. 15.1.3, that is blood group. As you know, blood of human being differ in certain aspects, though it appears to be similar. Various types of blood grouping of has been done. Two such groupings, that is the ABO and RH, are widely used all over the world. 15.1.3.1, ABO blood grouping. ABO grouping is based on the presence or absence of two surface antigens, that is chemicals that can induce human immune response on RBC, namely A and B. Similarly, uh, the plasma of different individuals contain two natural antibodies, that is proteins produced in response to antigens. The distribution of antigen and antibodies in the four groups of blood, that is A, B, AB, and O, are given in table 15.1. You probably know that during blood transfusion, any blood cannot be used. The blood of a donor has to be carefully matched with the blood of a recipient before any blood transfusion to avoid severe problems of a clumping, that is destructions of RBC. The donor's compatibility is also shown in table 15.1. Blood group A, antigen on RBC, A, antibody in plasma, anti-B, donor group A and O. Blood group B, antigen on RBC B, antigen in plasma, antibody in plasma anti-A, donor group B and O. Blood group AB, antigen on RBC A and B, antibodies in plasma nil, and donors group AB A B O. Blood group O, antigen on RBC nil, antibodies in plasma anti A and anti B, donors blood group O. From the above mentioned table, it is evident that group O blood can be donated to a person with any other blood group, and hence O group is individual are called as universal donor. Persons with AB blood group can accept blood. Persons with a AB as well as the other groups of blood. Therefore, such a persons are called universal recipients. 15.1.3.2, RH grouping. Another antigen, the RH antigen, similar to one present in a rhesus monkey, that is hence RH, is also observed on the surface of RBC of majority, nearly 80% of human. Such individuals are called RH positive, that is RH positive, and those in whom this antigen is absent, that is RH negative. An Rh negative person, if exposed to Rh positive blood, will form a specific antibodies against the Rh antigen. Therefore, Rh group should also be matched before transfusion. A special case of Rh incompatibility, that is mismatching, has been observed between Rh negative blood of pregnant mother with Rh positive blood of the fetus. Rh antigen of the fetus do not get exposed to the Rh negative blood of the mother in the first pregnancy, as the two bloods are well separated by the placenta. However, during the delivery of the first child, there is a possibility to exposure of the maternal blood to small amounts of the Rh positive blood from the fetus. In such cases, a mother starts preparing antibodies against Rh antigen in her blood. In case of her subsequent pregnancy, the Rh antibodies from the mother, the Rh negative, can leak into the blood of the fetus, that is Rh positive, and destroy the fetal RBCs. This could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called as erythroblastosis fetalis. This can be avoided by administering Rh negative antibody to the mother immediately after the delivery of the first child. 15.1.4 Coagulation of blood. You know that when you cut your finger or hurt yourself, your wound does not continue to bleed for a long time. Usually, the blood stops flowing after some time. Do you know why? Blood exhibits coagulation or clotting in response to an injury or trauma. This is a mechanism to prevent excessive loss of blood from the body. You would have observed a dark reddish brown scum formed at the site of a cut or an injury over a period of time. It is a clot or coagulum formed mainly of a network of threads called fibrins, in which dead and damaged formed elements of a blood are trapped. Fibrins are formed by a conversion of inactive fibrinogens in the plasma by the enzyme thrombin. Thrombins in turn are formed from another inactive substance present in the plasma called prothrombin. An enzyme complex an enzyme complex thrombokinase is required for the above reaction. This complex is formed by the series of a linked enzyme reaction, that is cascade process, involving number of factors present in the plasma in an inactive state. An injury or trauma stimulates the platelets in the blood to release certain factors which activate the mechanism of coagulation. Certain factors released by a tissue at the site of injury also can initiate coagulation. Calcium ions play a very important role in clotting. 15.2 Lymph tissue fluid. As the blood passes through the capillaries in tissue, some water along with many small water soluble substances move out into the spaces between the cells of tissues, leaving the large proteins and most of the formed elements in a blood vessels. This fluid released out is called as the interstitial fluid or tissue fluid. It has the same mineral distribution as that in plasma, exchange of nutrients, gases, etc. between the blood and the cells always occur through this fluid. An, an elaborate network of vessels called the lymphatic system collects this fluid and drains it back to the major veins. The fluid present in the lymphatic system is called lymph. Lymph is colorless fluid containing specialized lymphocytes which are responsible for the immune responses of the body. Lymph is also an important carrier for nutrients, hormones, etc. Fats are absorbed through lymph lactate in the lacteals present in the interstitial villi. 15.3 Circulatory pathways. The circulatory patterns are two types: open or closed. Open circulatory system is present in the arthropods and molluscs, in which blood pumped by the heart passes through large vessels into open spaces all over body cavities called sinuses. Annelids and chordates have a closed circulatory system in which blood pumped by the heart is always circulated through the closed network of blood vessels. This pattern is considered to be more advantageous as the flow of a blood or fluid can be more precisely regulated. All vertebrates possess muscular chambered heart. Fishes have two-chambered heart within the atrium and a ventricle. Amphibians and the reptiles, except crocodile, have a three-chambered heart with two atria and a single ventricle, whereas crocodile, birds, mammals possesses four-chambered heart with two atria and two ventricles. In fishes, the heart pumps out deoxygenated blood which is oxygenated by the gills and supplied to the body parts from where deoxygenated blood is returned to the heart, that is single circulation. In amphibians and reptiles, the left atrium receives oxygenated blood from the gills or lungs or skin, and the right atrium gets the deoxygenated blood from the other body parts. However, they get mixed up in a single ventricle which pumps out mixed blood, that is incomplete double circulation. In birds and mammals, oxygenated and deoxygenated blood received by the left and right atria respectively pass on the ventricles of the same side. The ventricles pumps it out without any mixing up, that is, two separate circulatory pathways are present in these organisms. Hence, these animals have double circulation. Let us study the human circulatory system. 15.3.1 Human circulatory system. Human circulatory system, also called the blood vascular system, cons- consists consists of a muscular chambered heart, a network of a closed branching blood vessels and blood, the fluid which is circulated. Heart, the mesodermally derived organ, is situated in the thoracic cavity in between the two lungs. Slightly tilted to the left, it has the size of a clenched fist. It is protected by a double-walled membranous back pericardium enclosing the pericardial fluid. Our heart has a four chambers, two relatively small upper chambers called atria and two larger lower chambers called ventricles. A thin muscular wall called interatrial septum separates the right and left atria, whereas a thick walled the interventricular septum separates the left and right ventricle. The atrium and ventricle are the atrium and the ventricle of the same side are also separated by a thick fibrous tissue called atrioventricular septum. However, each of these septa are provided with an opening through which the two chambers of the same side are connected. The opening between the right atrium and the right ventricle is guarded by a valve formed of three muscular flaps or cups. The tricupsed wall, whereas the bicupsed or metal wall guards the opening between the left atrium and the left ventricle. The opening of the right and left ventricle into the pulmonary artery and aorta respectively are provided with the semilunar valve. The valves in the heart allows the flow of blood only in one direction, that is from atria to ventricle and from ventricle to the pulmonary artery or aorta. These valves prevent any backward flow. The entire heart is made up of cardiac muscle. The walls of a ventricle are much thicker than that of atria. A specialized cardiac muscular cord nodal tissue is also distributed in the heart. A patch of this tissue is called in the right upper corner of the right atrium called sinoatrial node, that is SA node. Another mass of this tissue is seen in the lower left corner of the right atrium close to the anterioventricular septum called atrioventricular node, that is AV node. A bundle of nodal fibers atrioventricular bundles, that is AV bundle, continues from AV node which passes through the atrioventricular septa to emerge on the top of the interventricular septum and immediately divides into the left and right bundle. These branches gives rise to minute fibers throughout the ventricular musculature of the respective sides and called as Purkinje fibers. The nodal musculature the nodal musculature has the ability to generate the action potential without any external stimuli, that is, it is auto-excitable. However, a number of action potential that could be generated mm, in a minute vary at different parts of the nodal system. The SA node can generate maximum number of action potentials, that is 70 to 75 per minute, and is responsible for initiating and maintaining the rhythmic contractile activity of the heart. Therefore, it is called as the pacemaker. Our heart normally beats 70 to 75 times per minute, average 72 beats per minute. 15.3.2 Cardiac cycle. How does the heart function? Let us make a look. To begin with, all the four chambers of a heart are in relaxed state. That is, they are in a joint diastole. As the tricuspid and bicuspid valves are open, blood from the pulmonary vein and vena cava flows into the left and right ventricle respectively through the left and right atria. The seminal valves are closed at this stage. The SA node now generates an action potential which stimulates both the atria to undergo a stimulus simultaneous contraction, the atrial systole. This increases the flow of blood into the ventricles by about 30%. The action potential is conducted to the ventricular side by the AV node and AV bundles from the bundle of His transmits it through the entire ventricular musculature. This causes the ventricular muscle to contract, that is ventricular systole. The atria undergo relaxation, that is diastole, coinciding with the ventricular systole. Ventricular systole increases the ventricular pressure, causing the closure of tricuspid and bicuspid valves due to attempted backflow of blood into the atria. As the ventricular pressure increases further, the seminal valve guarding the pulmonary artery, that is right side, and the aorta, that is left side, are forced open, allowing the blood in the ventricular to flow through these vessels into the circulatory pathways. The ventricles now relax, that is ventricular diastole, and the ventricular pressure falls, causing the closure of seminal valves, which prevents the backflow of blood into the ventricles. As the ventricular walls declines further, the tricuspid and bicuspid wall are pushed open by the pressure in the atria exerted by a blood which was being imped, im- imparted, emptied into them by the veins. The blood now once again moves freely to the ventricles. The ventricles and atria are now again in the relaxed, that is joint diastole state as earlier as earlier. Soon the SA node generates the new action potential, and the events described above are repeated in the sequence and process continues. These sequential events in the heart, which is clinically repeated, is called as cardiac cycle, and it consists of a systole and diastole of both the atria and ventricles. As mentioned earlier, the heartbeat 72 times per minute, that is that many cardiac cycles performed per minute. From this it could be deduced that the duration of a cardiac cycle is 0.8 seconds. During the cardiac cycle, each ventricle pumps out approximately 70 ml of blood, which is called stroke volume. The stroke volume multiplied by the heart rate, that is number of beats per minute, gives rise to cardiac output. Therefore, the cardiac output can be defined as the volume of blood pumped out by each ventricle per minute and average 5,000 ml or 5 liters in a healthy individual. The body has the ability to alter the stroke volume as well as the heart rate and thereby the cardiac output and thereby the cardiac output. For example, cardiac output of an athlete will be much higher than that of ordinary man. During each cardiac cycle, two prominent sounds are produced which can be easily heard through the stethoscope. The first heart sound is lub, is associated with the closure of tricuspid and bicuspid wall, whereas the second sound is a dub, is associated with the closure of a semilunar wall. These sounds are clinical diagnosis significance. 15.3.3 Electrocardiogram ECG. You are probably familiar with this sense from a typical hospital television show. A patient is hooked up a monitorized machine that shows voltage traces on a screen and makes a sound pip 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 as the patient goes into cardiac arrest. This type of a machine, electrocardiograph, is used to obtain the electrocardiogram (ECG). ECG is a graphical representation of electrical activity of the heart during cardiac cycle. To obtain a standard ECG as shown in the figure 15.3, a patient is connected to the machine with a three electrical leads, one to each wrist and one and to the left ankle, that continuously monitor the heart activity. For detailed evaluation of the heart's function, multiple leads are attached to the chest region. Here we will talk only about standard ECG. Each peak in the ECG is identified with a letter from P to T that corresponds to the specific electrical activity of the heart. The P wave represents the electrical, that is excitation or depolarization of the atria, which leads to the contraction of both atria. The QRS complex represents the depolarization of ventricles, which initiates the ventricular contraction. The contraction starts shortly after Q and marks the beginning of the systole. The T wave represents the return to the ventricle from the excited to normal state, that is repolarization. The end of the T wave marks the end of systole. Obviously, by the counting the number of QRS complexes that occur in a given time period, one can determine the heartbeat rate of an individual. Since the ECGs obtained from different individuals have a roughly the same shape for a given little configuration. Any deviation from this shape indicates a possible abnormality or disease. Hence, it is a great clinical significance. 15.4 Double circulation. The blood flows strictly by a fixed route through blood vessels, the arteries and veins. Basically, each arteries and veins consists of a three layers: an inner lining of a squamous endothelium, the tunica intima; middle layer of a smooth muscles and elastic fibers, the tunica media; and an external layer of a fibrous connective tissue which collagen fibers, the tunica externa. The tunica media is comparatively thin in the veins. As mentioned earlier, the blood pumped by the right ventricle enters the pulmonary artery, whereas the left ventricle pumps the blood into the aorta. The deoxygenated blood pumped into the pulmonary artery is passed on to the lungs, from where the oxygenated blood is carried by the pulmonary veins into the left atrium. This pathway constitutes the pulmonary circulation. The oxygenated blood entering the aorta is carried out by the network of arteries, arterioles, and capillaries to the tissues from where the deoxygenated blood is collecting by the system of veins, and vena cava and emptied into the right atrium. This is the systematic circulation. The systematic circulation provides the nutrients, O2, and other essential substances to the tissues and takes the CO2 and other harmful substances away for elimination. A unique vascular connections exist between digestive tract and liver called hepatic portal system. The hepatic portal vein carries blood from the intestine to the liver before it is delivered to the systematic circulation. A special coronary system of the blood vessels is present in our body exclusively for the circulation of the blood to and from the cardiac musculature. 15.5 Regulation of cardiac activity. Normal activities of the heart are regulated intrinsically, that is, auto-regulated by specialized muscle, that is, nodal tissue. Hence, the heart is called myogenic. A special neural center in the medulla oblongata can modulate the cardiac functions through the autonomous nervous system ANS. Neural signals through the sympathetic nervous system, that is part of ANS, can increase the rate of heartbeat, the strength of a ventricular contraction, and thereby the cardiac output. On the other hand, parasympathetic neural signals, another component of ANS, decrease the rate of heartbeat, speed of conduction of the action potentials, and thereby cardiac output. Adrenal, adrenal medullary hormones can also increase the cardiac output. 15.6 Disorders of circulatory system. High blood pressure, hypertension. Hypertension is the term for blood pressure that is higher than normal 120 by 80 in this measurement 120 mm of Hg, that is millimeters of mercury pressure, is the systolic or pumping pressure, and 80 mm of Hg is the diastolic or resting pressure. If repeated checks of a blood of an individual is a 140 by 90, 140 over 90 or higher, it shows a hypertension. High blood pressure leads to heart diseases and also affects vital organs like brain and kidney. Coronary artery disease CAD. Coronary artery disease, often referred to as atherosclerosis, affects the vessels that supply blood to the heart muscle. It is caused by the deposition of calcium, fat, cholesterol, and the fibrous tissues, which makes the lumen of the arteries narrower. Then angina. It is called angina pectoris. A symptom of acute chest pain appears when no enough oxygen is reaching to the heart muscle. Angina can occur in a man and woman of any age, but it is more common among the middle-aged and elderly. It occurs due to conditions that affects the blood flow. Then heart failure. Heart failure means the stage of a heart when it is not pumping blood effectively enough to meet the needs of the body. It is sometimes called congestive congestive heart failure because congestion of the lungs is one of the main symptoms of this disease. Heart failure is not same as a cardiac arrest, that is when the heart stops beating, or heart attack, that is when the heart muscles is suddenly damaged by an inadequate blood supply. Summary. Vertebrates circulate blood, a fluid connective tissue in their blood, to transport essential substances to the cells and to carry the substances from there. Another fluid, lymph, that is tissue fluid, is also used for a transport of a certain substances. Blood complex a fluid matrix plasma and formed elements. Red blood cells, that is RBC, erythrocytes, white blood cell WBC, leukocytes or platelets, that is thrombocytes, constitute the formed elements. Blood of human are grouped into A, B, AB and O systems based on the presence or absence of two surface antigens A, B on the RBCs. Another blood grouping is also done based on the presence or absence of another antigen called Lysias factor, that is RH, on the surface of RBC. The space between cells in the tissue contains a fluid derived from blood called tissue fluid. This fluid, called lymph, is almost similar to the blood except for the protein content and formed elements. All vertebrates and a few invertebrates have a closed circulatory system. Our circulatory system consists of a muscular pumping organ heart, a network of vessels and a fluid blood. Heart has a two atria and two ventricles. Cardiac musculature is auto-excitable. SA node generates the maximum number of action potential, that is 70 to 75 times per minute, and therefore it sets the pace of the activities of the heart. Hence is called pacemaker. The action potential causes the atria and when ventricles to undergo contraction, that is systole, followed by their relaxation, that is diastole. The systole forces the blood moves from the atria to the ventricle and to the pulmonary artery and the aorta. The cardiac cycle is formed by a sequential events in the heart which is cyclically repeated and is called cardiac cycle. A healthy person shows 72 such cycles per minute. About 70 ml of blood is pumped out by each ventricle during a cardiac cycle and it is called the stroke or a beat volume. Volume of blood pumped out by each ventricle or a heart per minute is called the cardiac output and it is equal to the product of stroke volume and the heart rate, that is approximate 5 liters. The electrical activity of a heart can be recorded from the body surface by using electrocardiograph and recording is called electrocardiogram ECG, which is a clinical importance. We have a complete double circulation, that is two circulatory pathways namely pulmonary and systematic are present. The pulmonary circulation starts by the pumping of a deoxygenated blood by the right ventricle which is carried to the lungs where it is oxygenated and returned to the left atrium. The systematic circulation starts with the pumping of a deoxygenated blood sorry with the pumping of oxygenated blood by the left ventricle to the aorta which is carried to all the body tissues and deoxygenated blood from there is collected by the veins and returned to the right atria. Though the heart is auto-excitable, its functions can be modulated by the neural and hormonal mechanism. So thank you. Here we complete the body fluids and circulation. आपको ये वीडियो कैसा लगा प्लीज़ कमेंट में बता देना और मैं और क्या इम्प्रूव कर सकता हूँ और कैसे कर सकता हूँ वो भी बता देना. थैंक यू फॉर वॉचिंग दिस वीडियो.